Hi, everyone. Thank you for joining us. Aaron and I are actually re-recording the webinar we did last night on the topic of experimental design for young scientists and their teachers. We had to deal with a little power outage, and for the sake of you, the viewer, we're condensing it, and you're going to get an even smoother webinar than we had last night. But thank you for making time. And without further ado, Aaron, please take it away and tell us how we can help our students design better experiments.
Thank you, Blake. Yeah, we're very excited to go over this topic. We love this topic, so we don't mind doing it again. I want to talk about how the design process of a experiment or a student independent research project can be done in a way that facilitates analysis. So everything I'm going to be talking about here is geared toward this, thinking about what your students are going to do downstream with their data, the graphs they're going to make, and the analyses that they're going to do. And a lot of this comes from experience in my classroom that was earned the hard way by doing it the wrong way at first, and having students stumble in analysis, and me as a teacher not quite knowing how to help them. And then some of this is stuff that I've learned along the way as a professional researcher working on my own work. So the outline for the session today, I want to emphasize three main points that are going to lead to better analysis, and they're going to help your students set up better experiments in a way that leads to better science overall. So point number one is going to be to encourage students to focus. And I really want to emphasize that sometimes smaller, more focused questions make for better science. The second point we're going to cover is called begin with the end in mind. And this is just thinking through what you're going to do with the data at the end of the experiment. So you're going to ask questions like, what are my variables? What is my unit of observation? What will my table or spreadsheet look like? And very importantly, what will the graph look like that I'm going to put in my lab report, on my science fair poster, in my presentation? What is that graph going to look like? And you want to think about that before you've collected any data at all. And then the final point is repeat often. And this is just making a plan for adequate replication in the face of what variation that you're likely to see in your data. So we're going to talk about why you want to repeat often and collect as much data as you can. So quick break from our regularly scheduled program here to just give you a quick bit of lizard biology that you're going to need to follow my example. For those of you who don't know, I was a lizard biologist. I studied natural selection in the wild in lizard populations in both the Bahamas and in Florida. And I'm going to use an example that was actually data collected in my classroom in Chicago, but using lizards. So we need to understand some of this lizard biology first. So some points I want to make here is, one, I want you to think about the egg environment and consider factors like temperature and moisture that can greatly affect the development of an embryo, because there are a lot of materials exchanged through the eggshell and the outside world. So the egg environment is important. That's sort of my first point. One of the reasons the egg environment is important is because it can affect traits of a hatchling lizard, things like body size or color or running speed, things that are very, very likely to ultimately affect whether an animal lives or dies. So the egg environment is going to affect the traits of an organism. Those traits are going to affect the chances of survival. And here's where we get to the main point. Because this is such an important chain of events leading to the ultimate fate of an organism, the maternal choice of nest site is a very important decision. And it's a decision that is likely to have biological consequences throughout the lifetime of that animal. And so this maternal choice of nest site is an important decision. Okay, so that's my background. The biology you need to know to understand the example we're going to work with today. Okay, so point number one on my tips for experimental design is encourage students to focus. Smaller questions often make for better science. So first, I want to give you an example of a question that is a good question that can stem from a place of intellectual curiosity. But this question of how do females decide where to lay an egg, it's just too big to answer with a single experiment, and it's far too big to address with a student research project. There's just a lot of things that are not determined in this question that are open-ended. You know, how do females, that covers all species on Earth that have females. So that's a very broad, you know, subset of topics. And then how do they decide where to lay an egg? Well, that's something that's likely to be decided by dozens, if not hundreds of different factors. So again, it's just too broad to address with a single experiment. So let's try to narrow this a little bit. And so in this process of asking students questions about their question, they might narrow this down and say, okay, not just all females, but how do specifically female brown anole lizards decide where to lay an egg? So we're getting a little bit closer, but I would say this question is still too big because it's pretty hard to test. There's so many different factors that could affect this decision of where to lay an egg. It's going to be hard for us to get to an answer. So take it one step further. Do female brown anole lizards prefer wet soil when deciding where to make a nest? Now we have a question that can be tested. We're focused on this one specific factor of soil moisture, and we're able to set up some kind of experiment with this one species and this one choice to see if there's a preference for wet or dry when deciding to make a nest. So let's see how this translates to a design. At the most basic level, even before we've thought about, you know, how we're going to build an experimental apparatus, we're just thinking about this in terms of a experimental diagram. And I strongly encourage making these kind of diagrams with students, or rightly having your students make diagrams like this when they conceive of an experiment. And so this is just at the simplest level. If we want to explore this question, we're going to need to present a female brown anole with something that's dry and something that's wet to see which they're preferring to lay an egg in. And then we're going to need to multiply this by some minimum number of replicates. And so this is at the most basic level. There's still a lot of questions to answer about how do we actually build this experiment, but this is the basic design or model for the experiment. And then go ahead, Blake.
What would a good minimum number of replicates be? I know there's no hard and fast answer, but if there's something that you had to use to help a student realize that maybe their research question wasn't narrow enough, what would you use there?
This is a good question, and you're definitely right. There is no hard or fast number of replicates. But the thing I would say is, can you get at least 20 replicates? And if you can't, that's fine. There's a number of different constraints on materials and time and things that might not allow you to get 20. But if you can't get to 20 replicates, I would think long and hard before adding a third choice or a third level to this factor of soil moisture. And so that's kind of my general rule of thumb when I coach science fair kids, really from all over, that I say, if you can't get to 20 replicates, can you just keep your experiment to, you know, 2 possible choices or 2 levels of this treatment? I think that's a good rule of thumb, is to think about that 20 replicates.
And am I reading the experimental diagram right then, that even with just the 2 choices of dry and wet times 20 replicates, you have 40 observations as a part of that experiment?
No, that would only give you 20 observations, and that's sort of a minimum. Certainly 40 is not so many that you would say, oh, I definitely should, that's overkill, I should add another treatment to this. 40 is a very, rational sample size. Now, obviously you wouldn't want to do, say, 40,000 or something like that. But yeah, this is just 20 data points would be generated if this experiment were repeated 20 times. And repeating it might be the kind of thing you could use the same dry and wet setup, and you'd repeat it 20 different trials with 20 different animals. Or this it could involve creating 20 different setups, and so that's another consideration, and is to what goes into thinking about the experimental setup before you decide to add complexity to it.
Got it.
Now, if you did want to do a slightly bigger design, and say you had a few levels of that factor of soil moisture, instead of just a dichotomous choice between dry and wet, you could do this in a way where you provide 5 different levels of a moisture treatment. Still thinking about these as 5 different categories of moisture, but then you need, you're multiplying these 5 setups of whatever is holding this, this moisture, and then that's times 20. So it's, immediately a larger setup, a bigger creation. There's a lot more logistics that go into building that up. And this is, starts to be, I guess this is not a full factorial design. This is really just, 5 by 1, 5 levels of that one treatment. But I also want to show with the next slide, why you need to think long and hard about that focused question. Because if you start adding a second question and you want your experimental setup to do double duty and answer more than one question, well, then you're gonna, exponentially increase the size of your experiment. So let me show you what I mean by that. So if you have 2 questions, so we have our original question about, is there a preference for wet soil when making a nest? But if we want to add in a second question, that means we might be interested in, like, do these lizards prefer to nest near plants? Well, now we've got a whole second treatment. We've got this plant treatment. And even if that plant treatment just has 2 levels, either the presence of a plant and absence of the plant, if we want to do that in the same experiment, we now have what's called a 5 by 2 factorial design, where each of those 5 moisture choices needs to get repeated twice, once with a plant and once without, both levels of that treatment. So you can see then each experimental setup is going to have 10 choices, and then we need to multiply that by some minimum number of replicates. And this is an experiment I actually did with my students in my classroom in Chicago, and this was by far the biggest lab experiment I've ever tackled with students. It's actually the biggest lab experiment I've ever tackled, period. Most of my work was field experiments. And what we did is we made an experimental setup like this 5 by 2 model. This is what it actually looks like in a 10-gallon aquarium. And in each of these, we put in 3 females, which were going to be supplying the eggs, and 1 male to make sure that those females were in fact laying eggs. And then we multiplied that by 20 cages. And, so what this looked like in a classroom, this is, once we move the lizards into a single cage. So these are the 3 females from 1 cage, but we repeated that times 20 cages. So you can see it makes a fairly big setup that's fairly resource intensive. It's certainly resource intensive in the amount of time that it took to search those nest boxes twice a week for eggs. But even thinking about things like each cage has its own individual heat setup, and we're recording temperature in each individual cage. So this is just pretty big logistics to do this 5 by 2. I would not recommend this project as a single student independent research project. This project, we really relied on all 150 of my students pitching in on this project to make it happen. So thinking about that, the best experiment is often, the best design is often the experiment that you can actually do in a way that answers the question. So just wanting to think that through. And if you don't have that kind of time or resources to devote to an experiment, then maybe you don't want to use the 5 by 2 design. Maybe you want to use the simple 1 treatment, 2 levels, test it, make a comparison. And so that's something to think about.
Now, Aaron, for this experiment, I think you guys held yourselves to a pretty high level of statistical rigor. How did you impress on the students, got a wide variety of different students, the necessity for being really narrow in the research question that you were asking, and making and being like precise throughout the whole experiment? So I could see a lot of students just getting really excited about the fact that they have 80 lizards that they're in charge of taking care of. They're all laying eggs. So how did you balance the excitement and maybe the tendency for students' attention to wander and kind of keep it focused on being really scientific.
Well, one of the things that was really fun about this year, and I'll get into this in our next webinar when we actually go through the story of this, and that webinar is going to be on communicating with data visually. But one of the ways we did that and sort of maintain that level of rigor in the data collection here was, from the start, our intention was to publish this work in a major scientific journal, and so that was the standard we set in data collection throughout. And the kids really bought into that idea from the first day of school when we introduced this experiment, in saying, if we do this right, we're going to get an answer to a question that no one in the history of the world has answered before, and that we're going to publish this in the scientific community. And so everything we have to do has to be to this high standard. And there's all sorts of things we did, such as randomizing the order of the pots within the cages every time we search for an egg, and randomizing the order of the glass aquaria tanks here throughout the week as the experiments went on. And so we do a number of things so that we would be able to pass the level of rigor needed for peer review, and it ended up in the journal Behavioral Ecology. But students bought into that, and I had a couple of my scientific mentors were skeptical that we would be able to do it. They would say things like, I don't know, I've tried research with undergrads and it's really tough to get them to pay attention to detail. But I would offer this as a challenge to that, to people who say that young students aren't capable of research at a high level, because these were high school students. And another thing I should say is, like, I did not teach at a ritzy high school. This is not necessarily the highest performing high school. We, at the time I was working there, we had about a 50% graduation rate. So we were able to conduct research at that level despite a lot of challenges that we had. So I will say it is possible to do this stuff at a high level of rigor with high school kids.
Great way to apply keeping the end in mind.
Yeah, definitely. Okay, so that's a great segue to this, begin with the end in mind. And so this is where you want to ask yourself all the questions you're going to have at the end of the experiment. You really want to think this through before you start collecting data, because these are the things that will cause you to refine your design. These are the things that will cause you to come up with better plans for data collection, or even collect variables you might not have been thinking about. So first question I think you should ask is, what are my variables going to be? What is my unit of observation? And unit of observation is a really technical statistical term, and I'll define that in a minute. But so for this experiment and this question, really the only variable we need to analyze is the soil moisture treatment. And in a simple design, this would be, the value is either wet or the value is dry. In the design we used, the value was one of 5 moisture treatments. It was either 0% moisture in the soil, 12.5%, 25%, 50%, or 75%. And then we also recorded the egg ID. And this feeds into this concept of unit of observation. And so unit of observation is, what is the smallest independent unit of data being collected or observation being made, where the value of that is not going to be affected by any other observations in the experiment. And I say that our unit of observation was the single egg, and that is how we conducted our analysis. But if, strictly speaking, our unit of observation could also be considered the aquarium. So in that sense, we had 20 tanks, and the proportion of eggs found in each of those treatments could have been collapsed to a single data point, but we would have had less replication. We only would have had 20 data points in that case. What we did statistically is we recorded data on which tank the eggs were collected in, and then we tested for a statistical effect of each tank. And when we detected no statistical difference, or no statistical effect of tank rather, then we eliminated that variable from the data set for future analysis. And we were able to analyze data at the level of the single egg. And what that means is that each row in our data set represented a single egg. And so that is whatever you decide on as your unit of observation, that's going to be represented by one single row in the data set. And then so that when you think about what's your variables, what's your unit of observation, then you have all the information you need to think about, okay, what is my spreadsheet going to look like? And I can't stress enough, do not skip this step. And this is, I've said this to early career PhD students. I have said this to sixth graders. I have said this to, middle and high school teachers. Just don't skip creating the data table before you start. At least you want to know what the column headers are. Obviously, you don't know what data is going to fill them in, but you want to know what this spreadsheet is going to look like. And remember, we had two variables here. We had egg ID and we had soil moisture, and each row represents a different egg in the data set. And so it's going to look like this. And this is called tidy format, which is very much facilitates sophisticated data analysis in a variety of computer tools. This is how your students are going to work with data in their future careers, whether that's in business, in science, in media. If they work with data, it's going to be in this format. And it looks like this, where data sets tend to be long. Each variable is its own column. And as you add observations, your data set just gets longer and longer. And then this is a really important exercise. Once you kind of know what your variables are, what your spreadsheet's going to look like, take that one step further and think about what are my graphs going to look like? And I am a strong advocate of graphs. I personally, it blows my mind if I see a published paper that only has tables without graphs, which you don't see very often at all these days. But have students think through, even just with a quick sketch, what is the data going to look like if your hypothesis is not supported? And this, something like this, where there's no clear preference in the number of eggs found in the different soil moisture. So the y-axis here is representing the total number of eggs found, and on the x-axis you have soil moisture, and you see the 5 treatments ranging from 0 to 75%. And if your hypothesis is not supported, you're going to see likely some random fluctuation in the height of these bars, but basically no clear preference in which soil moisture the females were laying eggs. If your data were supported, you can look at this kind of a student sketch, and it's going to show, okay, we're going to see some kind of clear trend. And now this isn't the only way it could look. It could, you could also see a pattern where maybe all of the bars are close to the same height and one stands head and shoulders above the rest. Or you could see a pattern like this, where there's a strong preference for the higher moisture, and so you sort of see the stepwise pattern. You could also see this in the other direction as well. Maybe they prefer really dry. That would also support, the hypothesis that lizards use moisture as a factor. I guess that's one thing I didn't really define the hypothesis you're testing here. And the null hypothesis is that, there is no preference for moisture. The alternative hypothesis is that there is some preference for moisture, whether it's high or low moisture. And this example is what it would look like if there was a preference for high moisture.
And Aaron, did you ever, when going through exercises like this with your students, did you ever find that doing this bit of ideation would bias their observations? Like if they were kind of looking for support for their hypothesis, or was it more, did it way outweigh the organizational aspect of the experiment to be able to do this first?
Yeah, that's a really good question. And I would say it should not bias it. However, humans are humans. I think the important thing for dealing with that bias is setting up data collection in a way where when you're actually recording the data, that that bias is prevented in that case. And so in that way, in the way this experiment was done, they're dumping out the pots that have the different soil moisture. So if they're dumping a 0% moisture pot, there either is or there isn't an egg in that pot. It's not really a judgment call. And so they're simply tallying up the numbers, and any one egg that was found, you're not necessarily seeing this pattern because we collected the eggs over a period of a few months. And then at the end, we're producing these graphs. So that is actually one tip I would say is you probably shouldn't get into making figures when your data aren't done being collected. It's probably best to save the graph making for the end, so you don't find yourself rooting for one kind of a data point over the other, and you're just, genuinely looking at the data to see what conclusions you can draw. That was a good question. So this is, I want to show you the actual data from that experiment. This is the result of that student collected data. I believe it was 41 eggs were collected, and you can see it closely matched that hypothesis being supported sort of generic graph. Also want to show another way you could make this same graph in Data Classroom. This is as a stacked bar chart, and I'm going to actually hop over to Data Classroom and show you how to make this, because this is a new type of graph that we've recently added to the app. And so if you've been using Data Classroom for a while, you might not have seen this yet. So I'm just going to stop my screen share for a second and hop over to Data Classroom and show you that screen share.
And just generally speaking, with the stacked bar chart, is one of the benefits just that you can more easily count the total population.
I'll show you what you can do with that. You can choose to show the bar as a proportion, so that it's always the same height, and then it's like percentages of the samples, or you can make it as a raw count. And let me just share my screen here. Okay, so you can see my screen right here, and I'm going to go to that data set. I'm going to search for it. I have made this data set public, so if you want to play with this data set, you can. I believe this has the word classroom in it. Yeah, Classroom Lizard Project Nest Data is the name of this data set, and I'm gonna. Sorry, I just clicked on the wrong data set. This is the problem with doing live television here. So I clicked on this data set, and
is this the one that Christine owns?
It might be. I'm not going to make any changes to it. Yeah. So this is the data that I was talking about. If I go over to the graph, and I had this made already, but I'll undo this, and just to show you, if you want to look at this soil moisture, and in the appearance menu, this is what the default will look like when you do this. And so to make this graph, I'm simply choosing the histogram frequency icon, and this is a frequency plot, not a histogram. I showed soil moisture, and it put this up. You can also go into the appearance menu, and bars histograms will be the top option when you have this. And if you choose stacked, it will build this kind of stacked bar chart where the thickness of a color band represents the number of eggs collected there. And this is as a raw number. You can also do this stacked as a proportion, and then it will make it where the y-axis is showing 100%, and then the thickness of the bar is the percentage of observations made. And I'm going to actually go ahead and change the label of the y-axis to reflect that. And so you can see like that, I've just changed that to percentage of eggs collected. So this is another way. This is basically graphically the rectangular equivalent of a pie chart. But I find, especially when you have a really clear result like this one, where the 50 and the 75% are very, very overrepresented, it's a nice chart to quickly see the, the result. Of course, if you go back to this kind of a chart, I think it still is pretty clear what's, it's very clear what's going on, but this just gives you an option in how you communicate with the data. Okay, and I'll stop the share and go back. Okay, so here we are again. This takes us to point number 3. So point number 2 was begin with the end in mind. Point number 3 is repeat often. Replication is really the backbone of good science. It's the reason we have statistics in the first place, is because we expect there to be variation in outcomes that we can't control as a scientist. But in spite of that variation, we can see patterns that either support or don't support a given hypothesis. And I wanted to show you, this is for a different question. So just we've jumped examples here. We're no longer dealing with that question of is there a moisture preference when females are laying an egg. But in this example that lends itself to explaining about replication a little bit better, the question is, does incubation treatment affect body mass at hatching? And what we did was we incubated some lizard eggs at a bunch of different incubation treatments, and you can see the body mass of the lizard at the time of hatching. So each of these data points represents a single hatchling lizard when it was fresh out of the egg. And if you were to look at this with only 15 replicates, you don't really see a clear pattern. You can see these 95% confidence interval. They're very much overlapping. And I'm gonna jump back over to Data Classroom in a different data set and show you how to, I'm going to show you how to use the exclude feature, but I'm also in the process going to show you that effect of what replication can bring to a data set. So give me just a second to pull up the data set that I need. And this was also data collected in that classroom lizard project. And this is the hatchling lizard data. And so now I'm going to share my screen with you again. Okay, so now you can see my screen again. And here you can see the data with 15 replicates. And in reality, we collected 41 eggs in this experiment, and I've excluded some of those. So I'm going to go back to the table view, and I'm going to show you how I'm going to highlight all of the data in the table. So that's what I'm doing right here. And then I'm going to uncheck the exclude box over here at the right. And now I've de-excluded all those excluded data points. And if I go back to the graph, you can see here's what that same experiment looks like with all 41 data points. And you can see if we had stopped collecting data at only 15 data points, the trend would not be clear at all. But with 15 data points, you can see that the trend for a larger body size when lizards are incubated at these higher moisture levels is pretty clear. And this is statistically significant. If we could run the ANOVA on this, I could actually show that. If we run this ANOVA, you can see that this brings a statistical effect. The p-value is less than 0.01. So it's highly unlikely to get data like this if in fact there is not a difference. And we can also run this post hoc test to see that there's a significant difference between the 75 and 25% moisture treatments, as well as between the 75 and 50. The 50% and 25% were statistically indistinguishable from one another. But this is a really nice, strong, clear result, and it would not have been obtained if we didn't have the scale to collect sufficient data to see this, because there's a lot of variation around those mean values. I'm pretty confident that the true mean for each of these treatments is somewhere in, somewhere around where that that little mean and confidence interval is placed on either of those. But it's also clear that there's a lot of variation that is not determined by moisture treatment. There's a lot that goes into that. There's, genetics and probably time and some factors from that have to do with the mother as well. So there's a lot of unexplored variables in there, but despite that variation, we can still see a clear effect here.
And ultimately the result of having taken the time to have a narrow focus, to begin with the end in mind, and have the time to make all those observations instead of just being constrained with the 15.
Definitely. And it was really cool. And I'll also say that, when at the time my class was conducting this experiment, we were being mentored by Dr. Dan Warner, who at the time was at Iowa State University. And, this was not something that in my 5th year of teaching, I think was when we did this experiment. I didn't have this kind of expertise in experimental design, certainly not in statistics and how to analyze data, but I learned a ton on that project because as Dan was coaching us, he was thinking through these things, and a lot of these principles, were things he had learned in a career as a PhD researcher, and we were able to benefit from that knowledge and institute a lot of those into the design of our experiment. So I'm going to go back to here. And so, yeah, this again, just brings us to that repeat often. So with that, I was wondering, Blake, I know we recorded questions for the audience. Are there any other questions we wanted to cover before we wrap up this recording session?
Yeah, well, the one big question that is almost unanswerable, but I think we had a good discussion about last night, was the appropriateness of direct instruction versus other methods of instruction with such a complex process as experimental design. We know it's kind of something that's like still undecided in terms of what the pedagogy is, but maybe we could talk a little bit about helping some of those students who might struggle initially to set up their own experiment with some kind of scaffolding to get them to the place where they're actually doing student-led inquiry.
Yeah, that's a great question. And there, I mean, you said this as well. There's not one right answer to that, right? How do you do this? When do you use direct instruction? When do you throw kids in the deep end and say, okay, go design an experiment? And I would say I tried that method in different school years, and you often don't get a very high quality product from a student when, without much scaffolding and without much experience, you say, design your own, come up with your own research question, design your own experiment to test it. That's actually a pretty high level skill, and I don't think most students are capable of that right away. What I found has worked well is to do a big experiment where you are the model scientist for them, show them how it is done, talk them through the thought process, model that behavior first, sort of a, I'm a firm believer in the I do, we do, you do model of teaching. And I think for lab sciences, that works really well. Do one experiment where you're largely driving it. Do another one where you're genuinely doing it with your students. Maybe you haven't worked out all the things about the design, and you're giving students a chance to really contribute to that. They're thinking about how to collect data, but you're at your team doing it together. And then only, once you've done that, and they've done a couple of experiments from design to analysis, then they can go ahead and start working on their own experiments. Another thing I'll say is something that I didn't do when I was in the classroom, but I would definitely do now, is I would have students work through some experimental design scenarios where they're going to work through what was the thought process that some scientists used, and then here's the data set. Now you analyze it and draw conclusions from it, and then you could compare those to the conclusions that the scientist originally came up with. And I think that's a really powerful way. I would recommend, if you're a data classroom user, a number of the ready to teach exercises would allow you to do that with sort of minimal prep on your own. You could take one of the data sets that's from a fairly recent paper in the literature. You could have students work through the scenario, and then they make the graphs, they do the statistical tests, they draw the conclusions. I think that's great practice to have them do that before they're designing their own experiments, because it'll give them a chance to sort of, see what the end is. And then when you tell them to begin with the end in mind on their own experiment, they'll be able to do it in a better way.
Totally. And hopefully it's over different iterations, they'll begin to develop that kind of data intuition to get better at asking precise questions, at visualizing the end and defining their variables, and then being comfortable carrying out a number of replications.
Yeah, absolutely. Absolutely.
Cool.
All right. Well, with that, I guess we'll say thanks for watching, and also say that we're going to do a little tidy data tutorial, and we'll post that on the same place you can find this video. But thanks for tuning in, and thanks for watching. We hope to see you again soon.
Thanks everyone for your time. Take care.